This is our third lecture,
Sorry.
and we're recording in progress. It's our third lecture over 3 years. As you know, Joel very sadly died in June 30th, 2022, after an 8-month battle with brain cancer. This series is our way to celebrate his passion for understanding how science, policy, and Judaism can come together to help improve our world, particularly with regard to climate change in our own actions. I'd first like to acknowledge, I'm not sure they're on or not, but I'll say their names anyway. Joel and Carlene's two sons, Corey and Trent, I think are online on Zoom, along with their wives, Katie and Bree. And our greetings to all of you online. I can't see any of them, including many who've had the privilege of working with Joel over really many years. Students, postdoctoral fellows, colleagues, welcome to Temple Bethel. As many of you know, Joel was an extraordinary environmental scientist right here at the University of Wisconsin. Our lecturer today, Bob Hamers, will talk a lot more about Joel's contributions in a bit. And many of you also know Joel because he was a really insightful and much beloved member of our weekly Torah study sessions, 9 a.m. on a Saturday. The intellectual contribution of your life, it's great. And Joel is also an original member of the climate action team at Bethel, led by our fearless leader and Temple president, Marta Karloff, who's also doing Zoom today. So thank you, Marta. Marta, by the way, I just want to put in a plug. Marta is helping to organize a all-day session on plastics and health at the Wisconsin Environmental Health Network right on March 7th. So just in a little while. So if you're interested, please talk to Marta afterward.
Or Susan.
Okay. Oh, or Susan. Okay, great. Okay, either one. So you have heard me talk before about Joel's tremendous insights into science policy and the Torah. And I want to tell you a little more, but Joel delivered a remarkable lecture right at the temple that he gave in August of 2020. In a series of lectures called Conversations on Global Crises. This link, by the way, is or will be on the Temple Bethel Pedersen lecture website. It's really an amazing lecture he gave. He led a discussion right during the midst of COVID-19, so we were all on Zoom. His words are prescient, opening up, so I wanted to recite a few of them for you. Joel begins this way, remember that we're in the middle of COVID-19. One may ask, why talk about climate change now? Don't we have enough on our plate? Why address the topic of climate change when we are trying to cope as a country with a public health crisis and the economic consequences of our pandemic, and while we are reckoning anew with racial inequities in our country? Joel says, the reason is that the unfolding climate crisis is not going to wait for us. It is not going to wait for us to care and reform our society. The pieces are already in motion. The physics, chemistry, biology, all interlinked in our climate system, appear to be indifferent to our plight and to the resolution of our social ills. And that is the origin of the Pedersen lectures. Joel then proceeds to discuss a book that he had read about conservation, water conservation in California. The book was written in 2014. Two fruits are mentioned in what he relayed to us: the apricot and the pistachio. It's 2014. The apricots have bloomed too early. The leaves are too red, and they all fall to the ground. At the same time, the pistachio male and female trees are out of sync. There is no pollen. You need winter to make nuts, the author says, and there has been no chill. And there has been no chill because it is getting warmer. So Joel then goes on to describe his two views of humanity: the despot model, those who exploit the earth for its resources, and the stewardship model, one that promotes sustainable practices, conservation, and the responsible use of the environment. I think you can guess which view Joel adopted. And he lovingly quotes in the second creation account in Genesis, he quotes, in this creation account where God formed humans from humus, from soil, and places them in the Garden of Eden to till it and to watch it. Now, as we heard from Joe Handelsman last year, maybe we shouldn't be tilling it. We should be no-tilling it, but we should be paying very close attention and to act. And either way, this stewardship model is a lot more sustainable than the choice to subdue nature and to have full dominion over it. So our Pedersen lectures are all stewards of the earth. Jamie Schauer of the air, Joe Handelsman of the soil, and our lecturer today, who will dive into the microscopic world. The Torah here is again prescient. And also in Genesis, God says to Abraham, I will bestow my blessing upon you and make your descendants as numerous as the stars of the heaven and the sands on the seashore. Now that's, and Abraham walks away. This is the Akedah. All right, I'm here to tell you that there's a missing passage here, and I'm going to tell it to you. What God really said to Abraham was, Abraham, he says, go forth to the seashore and touch the sands. So Abraham goes to the seashore and he's staring at the beach. And God says, Abraham, look more closely. And Abraham bends down and God says, the grains of sand, they are really tiny. Touch them. They are created by the stars of the heavens. And then God reveals all. He says, 4,000 years from now, institutions called the National Science Foundation and the National Institutes of Health will freely and without restrictions, unless momentarily, fund research to explore scientific questions involving these nanoparticles, these grains of sand, both for their potential health benefits and their harmful benefits, ultimately to make the lives of future generations healthier and more sustainable. Abraham, of course, had no idea what God was talking about. But he snatches up a pile of sand in his hands, and it falls through his fingers and into the beakers of today's research labs. Abraham was, alas, a few thousand years too early, so it falls on our lecturer today in the Center for Sustainable Nanotechnology, which he leads, to carry out this mission on our planet. Our Pedersen lecturer is Bob Hamer, scientist extraordinaire, who will take us on a journey through nanotechnology. Bob has many accolades as leader of the chemistry department in many efforts across campus, as co-founder of the company Cybertronics, as director for the Center for Sustainable Nanotechnology, and as a community leader who, along with colleague Mark Ediger in the back, created a program to bring underrepresented minorities to campus called the Bridge to the Doctorate. Here, I think I've mentioned every word that I shouldn't have mentioned. Okay. There is one word I think that best describes Bob, but it is not actually found literally in the Torah. It has its origins, I think, and I looked it up in Cicero's, actually, Humanitas, written about 60 BCE. It's the Roman version of education and civic virtue. Humanitas was translated into German much later as, I get this right, Menschlichkeit, in around 1860, was truncated to describe a person, of course, a Mensch. A good and honorable person of integrity and compassion. Many people helped Joel in his last year, and many of them are online. But I think no one in Madison was a greater friend to Joel. During his last year, helping Joel and his students in ways that could only be translated as the Hebrew word chesed, meaning loving kindness, a word that abounds in the Torah. So it's our great honor for all these acts, personal and global, to welcome Bob Hamers as the 2025 Pedersen Lecturer and to kudo him in the environment as one who has truly contributed to repairing the world. Please join me in welcoming Bob to Temple Bethel.
Thank you. Can you go in full presenter mode instead of seeing most of it?
Oh, Let's see, I always have to remember how to do that. Swap displays, I
There
think
you
that's
go.
it.
Perfect.
Yep.
Thank you.
Okay.
Excellent.
One thing I have to learn is never try to follow a Gail on a. Well, thanks for inviting me. It's a real pleasure to be here. And I'll tell you a little bit about today, some of the work that Joel and I have done together. Given the, broad nature of expertise in the audience, I'm gonna try to get a nice balance between giving you a little bit of detail on what we do within the center and what Joel has done. So we'll see how that goes. So anyway, there's a photograph of Joel and some of my colleagues. Gil mentioned the Center for Sustainable Nanotechnology that I direct. And Joel was a big part of that, and a couple of our colleagues, Christy Haynes and Rebecca Clapper. Okay, so our focus is on
No.
nanomaterials. They're all on the same page. What are nanomaterials? So there's a definition of nanomaterials. That's an official definition, which is a material that is something that is nominally a solid that has at least one dimension that's less than 100
Bob,
nanometers.
excuse me, do you know how to switch this so we see the full screen and not the next?
People are seeing the full screen, but here we're somehow seeing a different view.
Maybe that's the best we can.
No, we should be able to see.
Oh, swap display. Can we go with
Well,
the
that's what I
swap
did.
displays?
Yeah, you have to choose presentation, not presentation with notes.
Uh-huh.
Let's see.
It takes a village.
See if you go back here.
Yeah, displays.
He's connected to that display.
Let's see.
No, swap displays will swap on the
Okay,
view.
the view, gotcha, gotcha.
Well, that's what I did.
Oh, you did that.
Yeah.
Yeah, try it again.
Right, okay, maybe it doesn't. Okay.
Oh,
Can
it's
you
always
get up
something.
the swap displays up at the very top? Okay, all right, I guess we're okay. Sorry, I didn't mean to.
I'm doing it exactly.
Okay, you should keep going. It's fine, we can
Yeah.
see it.
Okay,
All right.
okay. Okay, so the, okay, let me just go back here for one
Sorry about that.
second. Oops, I'll go back. Okay, so a lot of people have a hard time like visualizing what is 100 nanometers, right? So a good benchmark is a human hair. So a human hair is about 100 times larger than 100 nanometers. So this just shows some blow-ups of what that might look like. Another good benchmark is, you'll mention that Joel was really interested in like the coronavirus. So that's a virus particle, and this is 20 nanometers across the 20 nanometers here. That little scale bar there is 20 nanometers. So this is about the size of kind of what becomes nano. Okay, so first off, I want to say there's many different kinds. We talk about engineered nanomaterials. So there's lots of nanomaterials that are naturally occurring in the environment. Sand, for example, the small particles of sand. There's particles of dust that are less than that. And we've kind of adapted to these, right? They're not hazardous to us, but we can now make them in many different chemical compositions and also many different shapes. So this is just an example. The top is a few things that one would generally think of as being non-toxic. So the one on the top left, iron oxide, that's rust. Okay, so nano rust, nobody ever died from rust. The one in the middle is zinc oxide, that's diaper rash ointment, okay? Titania is present in lots of different things, it's present in foods. But then we get to materials that are being engineered now, or maybe because of the different chemical compositions and shapes, maybe we do have to think more about whether these materials might be potentially hazardous either to humans or to the environment. So I'm going to talk a lot today, actually, about this material right down here, lithium cobalt oxide. So this is a material that's used, all of you who have cell phones, right? There's lithium cobalt oxide in your cell phone, in your laptop. If you have an automobile, the electric vehicle batteries have a large amount, like 100 pounds of this material, roughly, in your car batteries. And then I'll talk later about some of these copper compounds. But I just want to point out that when we talk about nanomaterials, there's chemical composition, size, and shape. And we can also make these in many different shapes, like round particles, rods, tubes. Down here, we have like little nano spikes, just so we can say, ouch, right? Pretty much, or little flat sheets. So there's a lot of variety, many different properties of nanomaterials beyond just size that we have to worry about. So I pointed out there's found in lots of different consumer products. So we talked a little bit about sunscreens, there's in paints. Titanium dioxide is a really white material that's used in paint, but it's also sometimes found in the glaze of donuts. Your catalytic converter of your car batteries, so they're all over, getting incorporated more and more into consumer products. And so then we have to also worry about what happens at the end of life of those products. Now you might ask, why nano? Like, why do people want to make things nano? And what is different about things that are nano? And I'll simply point out that when you make things small, when you have things that depend on the surface area, that is the outer periphery. Think, for example, of a chemical reaction that might be taking place on the surface of a solid particle. If you take the same amount of material and you carve it up into little tiny particles, you actually get more surface area for the same total amount of material. So that's one of the big driving forces for nanomaterials, is simply getting more surface area for the same amount of material. That's actually good, right? That usually enhances sustainability if we can do more with less. So again, a couple examples. So your catalytic converter is one of the earliest examples of something that incorporated nanotechnology into it. The catalytic converter of your car converts carbon monoxide and nitric oxide, both of which are toxic, into carbon dioxide, which is, we have problems with CO2, but it's a lot better than carbon monoxide, and nitrogen, which is 78% of the atmosphere anyway. It does that by having a ceramic matrix and then putting little tiny particles on that are very small, just a few nanometers in diameter. And what I call the great conspiracy of the periodic table is that oftentimes the elements that are most useful are those that are most rare. And in your catalytic converter, platinum, iridium, rhodium, if you look up, like these are like the three least abundant elements in the periodic table, pretty much. And so we want to make the most effective use of them. And so by dispersing them into little teeny tiny particles, you actually enhance the sustainability simply by doing more with less. And this has been around, this technology has been around probably since the 1970s would be my guess. So in the catalytic converter, we use nanomaterials because they provide specific chemical properties that we need, and also cost. You'd like to reduce the amount of material that you use partially for the cost. But there's other reasons why people are interested in nanomaterials. Sometimes it can be the optical properties. So little tiny particles often will have unique optical properties that can be used in things like biomedical imaging. So that's, again, as well as scientific imaging that's non-medical. But here's an example of a rat that's been injected with a little tiny nanoparticle. Actually, with some kinds of nanoparticles, we can change the optical properties just by changing how big it is. Now, there's some of this is going back thousands of years with gold nanoparticles. I think it was the Romans who discovered that you could make colored coatings using little tiny gold nanoparticles. But this can be broadened to other kinds of things that we can make in large quantities, and they're made from inexpensive materials like carbon, for example. One of the things my center has done a lot of is focusing on making these kinds of brightly luminescent particles simply using carbon. And then finally, if we come to an area like energy storage, lithium-ion batteries, like many of us believe that going to transition to electric vehicles is actually environmentally beneficial. It provides much less emissions of smog into the atmosphere. And in this case, we have things that are used because of their mechanical and electrical properties. So inside your lithium-ion battery on both the positive electrode, which is called the cathode, and the negative electrode, which is called the anode, you have little tiny particles of materials that absorb lithium. And that's, they shuttle lithium back and forth between the anode and the cathode. By making those particles very small, you increase the ability to charge and discharge your battery faster. So as everybody wants an electric vehicle, especially that can be charged quickly, the way you achieve that is by making the particles really, really small. Okay? Again, you have more surface area, more things can happen to make it charge. All right, so there's many different reasons why people want to use nanomaterials. And again, these are examples of what I'll simply call engineered nanomaterials as distinct from naturally occurring nanomaterials. So the question that Joel and I have been working on for the last 20 some years, and is the focus of my center is, okay, so we take these materials and we engineer them to be useful in some particular application. That's great. What happens to them when it's done? We do live in kind of a throwaway world, right? So what happens to those materials when they get somehow into the environment, either intentionally or unintentionally? So there's lots of things that happen. I'm not going to go into great detail on this, but we know that they can undergo various kinds of chemical reactions. Those chemical reactions can release things into the environment that might be potentially hazardous. This is a really complex problem because once they're in the environment, there's lots of things that can happen. They can also get coatings. So in a natural environment, there's things like degraded organic material or so-called NOM, naturally occurring organic material, that can adsorb to the surfaces of these. That controls everything that happens to them. So it's a very, very complex problem that we're trying to understand. And we need to understand that if we really want to be able to predict and understand what is the environmental impact of nanomaterials if they potentially get released into the environment. Another aspect of this, and actually where Joel placed a lot of his focus, I'm a materials guy, I'm not a biology person. I'm a person who makes nanomaterials and characterizes them. Joel, because of his background in environmental science, was very much interested in understanding more of the organism side. What happens to those nanomaterials when they interact with different kinds of organisms? And in fact, a lot of Joel's interests were focused on understanding, if we want to understand the impact of a nanomaterial on some kind of biological organism, the interaction of that nanomaterial with the cell membrane. So cells always have a membrane, or in a plant, it's a cell wall, but there's always something that defines the outer periphery of the cell. And maybe we can understand a lot about what happens simply by trying to understand that very first line of interaction, and the fact that different kinds of cells, for bacteria, gram-positive, gram-negative, vertebrate, invertebrate cells, plants, all have different kinds of chemistry on the outside of those cells. And maybe we can think about this environmental impact as a chemistry problem. And in fact, the chemistry division of the National Science Foundation has funded our work because they wanted us to focus on understanding this very basic fundamental interaction of the nanomaterials with the biological organisms. So all these different kinds of cells have very distinct kinds of chemistry on their surfaces. And so this is really where Joel spent a lot of his time thinking about and trying to understand. So again, there's many things that can happen along the way. So some kinds of nanomaterials can release their components. So something like lithium cobalt oxide can release cobalt into the environment. Cobalt is a relatively toxic metal. They can produce what are called reactive oxygen species. So you all know that radicals in generally are not good, right? In terms of like exposure to life forms, right? Radicals generally are bad. So there's a class of radicals called reactive oxygen species that are naturally produced, that are something we look at. And in general, just understanding these interactions with the organisms is kind of where Joel was. When we think about this problem, we kind of can go back many years and think about examples where there have been technologies that have been developed and scaled up and implemented on full scale, where it was only years later, we actually discovered there were environmental impacts. And I can go all the way back to DDT back in the 1960s. When small amounts of DDT in the environment were causing thinning of bird shells, and we almost saw the bald eagles become almost extinct. The ozone hole, there have been examples of endocrine disruption in fish due to synthetic androgens. And, just the last few years, we've seen now a lot of interest in PFAS, perfluorinated alkyl substances, two of which I've shown examples here. These are materials that are nominally non-reactive. Okay, from a chemistry standpoint, fluorinated carbons are like the most stable kinds of compounds you can imagine. We never dreamed that they might accumulate in tissues and that that accumulation might actually cause some adverse health effects. So the important point is this is kind of a long-term thing we need to be thinking about is even if nanomaterials might not be like acutely toxic in release in high concentrations, there's also a question about what happens over long-term, low-level kinds of exposure to different kinds of organisms, or where various kinds of accumulation might occur in either in sediment or other kinds of natural environments or inside different kinds of organisms. Our work has been really focusing on kind of the chemistry side and the idea that if you want to understand and predict this kind of thing and prevent it in advance, what you'd really like to do is understand what's happening and develop a predictive ability so that you can predict in advance what happens if certain kinds of nanomaterials were to get released into the environment. That's a hard problem, okay? There's a lot going on, but it's why, in fact, this kind of research needs people with all different kinds of backgrounds, all working together at the same time. People who are interested in the making of nanomaterials, characterizing them, characterizing the biological interactions at a molecular level, people who do toxicology, and people who are on the engineering side who understand like what are the next generation of nanomaterials that will be coming up. Okay, so this is kind of where we are. So this is just a really complicated problem because lots of things can happen. We can have nanomaterials going into a cell. Okay, once they're inside a cell, they can undergo chemistry and release toxicants, for example. We know that nanomaterials don't have to get into a cell in order to have impact. They can, for example, they can dissolve, they can transform, they can release ions, they can do things on the outer periphery of the cell. They can produce these things I call reactive oxygen species, radicals, generally not good for you. And much of Joel's interest was on this understanding of this, how do nanomaterials bind to cells and what is it that controls that? Okay, so it's a complicated problem. I'm going to show a couple of slides that Joel himself made. This is one example. This is a slide that Joel made in describing his own group. And I simply pointed up because Joel is really uniquely suited to tackle the kinds of problems that I'm talking about. Joel, in our center, had one thing that none of us, the other 15 people in the center, really had, which is credentials as an environmental scientist. So the rest of us were chemists, like trying to apply our knowledge to environmental problems. But Joel, because of his background as a real environmental engineer and experience actually working in that field, was able to bring really unique insights and actually to keep us on track. Because as chemists, sometimes we want to go over here. And Joel was always, well, to rein you back in over here. So Joel was interested in a lot of his early work focused on things like organic contaminants in the environment. His tenure case was largely based on his work in prions, or transmission of neurodegenerative diseases. These kinds of things relate directly to understanding what happens to our nano-environmental interactions. And of course, Joel was interested in making things better and what are called advanced treatment processes. So he and I had a collaborative grant for a number of years trying to take this technology and relate it to advanced water purification. Now, I'm not going to talk about that today. There's already way too much to talk about. So, okay, so how did Joel and I meet, right? So it turns out that back around 2004, I became the associate director of a center. So a center, when I talk about a center, I usually mean a large collaborative research grant. It's not a physical center. It's not a room or a building. It's simply a framework for people to collaborate. And there was a center on campus that I was the associate director of, second in command, called NSEC, the Nanoscale Science and Engineering Center. And this center was focused on nanomaterials related to microelectronics. And in the context of that center, I was doing work in collaboration with other people, taking some of these nanomaterials, these are some of these luminescent nanomaterials, and applying them to advanced technologies for microelectronics fabrication. And you probably can't see it in this image, but there's little nanoparticles lined up in rows along this direction. It's not intended to look like a flag. It's intended, that's just a blow up. But the important point is we were looking at nanomaterials for microelectronic applications. This center had 4 research thrusts, complementary research areas that all kind of linked together. And this is one on drug assembly, is how do we control the way that nanomaterials assemble into larger objects. And there was also a thrust on what was called societal implications of nanotechnology. And it turned out that the person who was in charge of this left the university. And so there was a vacuum. We had this funded grant and we were supposed to be doing work in this area, and the person left. So what are we gonna do? Well, we issued what was called a seed proposal. We sent out a call and said, people who are interested in this area of societal implications of nanotechnology, send us your ideas, and maybe we will pick up on some of them and formulate a new thrust for it. Well, it turns out that Joel responded to that and submitted a proposal. And at the same time, another scientist named Dick Pedersen, spelled differently, Dick Pedersen was a toxicologist who worked with zebrafish as a model for understanding toxicology. They both submitted proposals in kind of the same area. So I decided, well, why don't the 3 of us work together? Because I can make materials that then the other 2 people in the collaboration can use. And that's how we met, right? That's how we started, and we started collaborating. The very first collaboration study that we did was looking at the toxicity of these cadmium selenide quantum dots. So these are the bright luminescent nanoparticles that I showed you a few minutes ago. These were being used at the time in laptop computer displays. They emit very pure, very bright red, green, and blue by changing the size of those particles. And so they were being used as replacements in laptop computers because they gave very pure colors. But again, the question was, well, what would happen to those laptop computers 3 years from now when they're no longer being used? All right, so I made the nano, and I didn't, my student made the nanoparticles. This is Drew Mangum. As you can see some of the green ones here. I'm not gonna go into how these are made. I do wanna point out that these nano, we worry about a lot when we're trying to understand these. So these nanomaterials often have several different layers. There's a core at the very center. Sometimes there's a shell around that, kind of a chemically protective shell. And then most of the time, there's some kind of organic fluff that are called ligands, okay? Ligands on the outside. And those are usually put there intentionally to try to control, like, does the nanomaterial disperse in water, or does it not want to disperse in water? In this particular case, these nanomaterials were coated with something called PEG, polyethylene glycol. Now we have to worry about the toxicity of all these different components. Many of you have experience with PEG because if you've ever had a colonoscopy or ever taken Miralax, you have ingested PEG. Okay, so it's not toxic, right? And that's an important part. Is the ligand itself is not toxic. So then we took our friend Dick Pedersen, who is an expert in zebrafish, would essentially make zebrafish to form embryos, and then those embryos would be exposed to these nanomaterials. Right? That's kind of how we would assess the toxicity of the materials. And then after some time, we would visualize and look and see, could we see any kind of external manifestation of toxicity that made it exposed zebrafish different from one that was not exposed? Okay, so here's an example from this very first study that we did together. And I'm not going to walk through detail in this, but here's a normal zebrafish. And then we coated these nanomaterials with different kinds of organic ligands on the outside. But you can see that all of these on the right-hand side are different from the one up here. They all have some kind of external manifestation of toxicity. And it might actually be different depending on the kind of coating. But if we simply said, well, what are the components? Cadmium, selenium. We can do control experiments where we say, suppose we take some other form of cadmium and expose the zebrafish to that. Is that toxic? And it turns out the answer is, well, a little bit, but not anywhere close to the kind of response that we're seeing here. So there's all these kinds of control studies we have to do to make sure that we've isolated what we think we're seeing is actually what we are seeing. So the important point from the study is that we showed that these cadmium selenide nanoparticles were much more toxic than any of the individual components, right? PEG is pretty much non-toxic, that external coating. Selenium itself is not particularly toxic, but what's happening then is the nanoparticles are getting inside the zebrafish, and then once they get inside, they can dissolve and release cadmium inside the organism. Whereas when we expose it to other forms of cadmium, the cadmium has to somehow get inside first, right? And we all have, our skin, right? Fish skins, those are good defense barriers against things. So it's almost like a Trojan horse, right? A nanomaterial can actually get inside the organism and then do damage after it's in. Now you might think, well, things that are toxic, well, maybe we expect this, but you might also say, well, things that we know or think are non-toxic can also have an effect. So I mentioned that titanium dioxide is used in coating on donuts, okay? It's used all over the place, and we don't usually consider it to be toxic. But if we take titanium dioxide in a nanoparticle form and expose zebrafish to TiO2, we see that there are all kinds of malformations of the zebrafish that are distinct from that of, well, actually, we see that there are malformations, but only if the lights are on, okay? Photochemistry. So photochemistry can do a lot of damage as well. That is, there are chemical reactions that can occur that have to have some kind of supply of energy, okay, in order for them to occur. So this is an example of something where sunlight interacting with the nanomaterial creates radicals inside the organism. And those radicals then actually do damage. So they can create, this is mortality as a function of the concentration. And this all occurs. These all occur because sunlight provides the energy needed to drive reactions that form toxicants inside the organism. Okay. So this was kind of where Joel and I started our collaboration. That collaboration ended, or that large grant ended around 2010, but it was just this little, our team of 3, right? And we had about 2 or 3 graduate students, and that was it. So as that was kind of coming to an end, Joel and I thought, we want to continue working together. We actually want to expand this effort and make it broader, more general. And so there was an opportunity from the National Science Foundation to create a new center. And so Joel and I have had this idea, let's broaden this and create this new center called the Center for Sustainable Nanotechnology. And so we've been funded since 2012 by that. That actually ends in August of this year. Can't be renewed further. But this is kind of where we've been going now. This center has many of the same goals as our original collaboration, except now it's bigger. So we've had at times as many as 15 faculty at about 12 different institutions around the country. So it's not just Madison now, it's actually a national effort now. And involves about 75 people altogether, about 40 PhD students, 15 faculty, a bunch of undergraduates, high school students, everything. So here's just a photograph of the team from one of our earlier meetings. And Joel was, you can barely see him way back there, but Joel was there. And okay, so when we founded this new center, we wanted to focus on understanding this nexus between nanomaterials and emerging technologies. That is, there's lots of different kinds of compositions of nanomaterials that one could worry about. And some of them are materials that might only be produced in very small quantities. And if something's only produced in very small quantities, you might question, is it going to be an environmental risk? Probably not. So we decided to kind of do an assessment of what are the materials that we should actually be looking at if we want to understand what's the environmental impact. And so we look at three things. One is how much material is being produced? Are there potential routes of exposure? And are the materials potentially toxic in themselves? So that we might have reasonable suspicion that there could be damage. And we came across in general the area of e-waste as being one that we really needed to look at. So electronic waste, and in particular, the kinds of materials that are going to things like lithium-ion batteries and other kinds of energy storage devices. Partially because of the materials, the toxicity, but also there's very little infrastructure. So almost none of the lithium-ion batteries that are being used today are recycled. It almost all goes into landfills. Now, a properly maintained landfill maybe releases little into the environment, but in practice, a lot of the e-waste that we generate in this country doesn't get landfilled here. It gets shipped off to other countries that have less stringent environmental standards, and other people then have the potential risk of being exposed to them. Okay, so we're going to focus a little bit more on these materials that are used in lithium-ion batteries and energy storage. So inside your laptop computer, if you were, or your cell phone or laptop, you would have this material called lithium cobalt oxide, or LCO. If you're driving an electric vehicle, you have between 75 and 100 pounds of this material called NMC, nickel, manganese, cobalt. Cobalt is very expensive and is a finite worldwide supply. So there's a lot of interest in replacing cobalt with other elements that are more abundant. And these include nickel, manganese, and cobalt. For various reasons, if you put all three of these in, you can actually make a material that has exactly the same crystal structure. It's basically the same material. You just kind of swap cobalt for maybe manganese, or maybe you swap two cobalts for one manganese and one nickel, and you make this material called NMC. So I'm not going to talk a lot about the details of those, but this is kind of the direction that we've been going. And when we did our first studies assessing the impact of these kinds of materials, we use a different organism. We use a bacterium called Shewanella oneidensis. It's a soil bacterium, so it's a good model system. If materials are not being properly maintained in a landfill, then it's the soil bacteria that will be exposed to these kinds of materials. So we choose an organism appropriate. And what we see again is that these heavy metals are being released. We see like nickel coming out. We see clear evidence for these radicals being induced, and we see that the bacteria are being delayed in terms of their development and or completely dying. So we see that they induce significant toxicity. So maybe a little bit of a warning sign. They can also do other things. So we worry a lot about what happens if a payload gets into an organism. We see direct evidence that DNA, or that exposure to these materials damages DNA. So here's an example of looking at a method called gel electrophoresis, and basically what it shows, or what we can learn from this kind of image, is that the strands of DNA are being broken. And we also see evidence that the strands of DNA can be modified. There's various kinds of modifications of DNA, many of which are important to actual life function. but many of them can be damaging and alter the way that the DNA behaves. So this again is something that we want to be a little bit concerned about. The center that I run, because, you know, while we're interested in the environmental aspects, one of the key challenges is with so many different kinds of materials that we can look at in terms of composition, size, and shape. Okay, so we kind of picked ones based on what we thought was happening with emerging technologies. There's also a question of, well, if you want to understand the environment, there's like a gajillion different organisms that you could study. You could study zebrafish, you could study bacteria. How are you ever going to test all these different kinds of organisms, right? It would be an overwhelming task to actually think about testing everything. So this is also where Joel's expertise comes in, is we really take a different approach in which what we're really trying to do is understand at a very fundamental level, what are the fundamental interactions that take place, with the idea that, for example, different cell types have different chemistries on their surfaces, different kinds of chemical functional groups. If we can understand something about the nature of the interaction, then we can do tests on a small number of species, but we can generalize that to a much broader class of species. So that's kind of the approach that we're taking. So that requires that we be able to connect all the way from very, very fundamental chemical reactions that are taking place, all the way up to what's happening inside the organism, right? That's a hard thread to draw. But actually we succeeded, largely in part to Joel and other people within the center. And here's just one example of a little bit of a thread, right? So we're looking for, like, what are general mechanisms by which nanomaterials interact with different kinds of organisms? So this is an example of a sandworm. It's called a chironomid. It's red. It's about a couple centimeters long, so it's about an inch long. It's something that's found in the bottom of stream beds. So again, if nanomaterials were released into the environment and if they somehow settled out, they would go into the bottom of the stream, right? Where these organisms might be exposed to relatively high concentrations, even if it was just small concentrations that were led into the environment. Kind of a kind of concentration effect. So this organism is red, okay? If you expose it to these kinds of nanomaterials, you see it suddenly, can you see the organism? It's there. It's transparent, okay? Here's a blow-up. It's smaller, but it's also transparent, okay? Well, what does that tell you? Like, why is blood red? Why is the organism red? Because almost all living organisms have heme proteins. Heme, as in hemoglobin, it's a form of iron, right? And it's what makes blood red. So when we see something like this, we can say, well, probably this nanomaterial is somehow impacting the way that heme is made inside the organism. So we can go down deeper and say, well, what's the mechanism by which heme is made? And there's different kinds of proteins. And one of them is this thing called a conatase. This is an enzyme. And in the center of the enzyme is a little cluster of 4 iron atoms and 4 sulfur atoms. And this actually is critical to actually making heme. We can actually look at that using advanced analytical techniques that I won't go into detail, but we can directly see the way that the nanomaterial is impacting this little cluster of 4 iron atoms and 4 sulfur atoms. So that allows us to draw a thread all the way from what happens chemically in terms of the way the nanomaterial interacts with the organism, all the way up to what is the external manifestation and impact on this organism. So that's one example, but it's kind of like, it's hard to explain, but it's kind of a hurrah moment for our kind of center, where we are trying to really draw a thread all the way from fundamental chemistry, all the way up to what happens in a complete organism. Another example is we can see nanomaterials interact with different kinds of molecules that we know are critical to what's called cellular respiration. So respiration is simply converting food into energy. the cellular respiration basically creates ATP, which is the energy molecule. And NADH is an integral part of that process of creating energy from food. And we can see that exposure of these nanomaterials to NADH actually, it decreases the NADH. It's basically reacting with the NADH. And through that process, it's impacting the electron transport chain. So again, another example where we have kind of a general mechanism that allows us to extend this beyond just a single nanometer. Okay. Now, one question you might say, all right, what I've focused on so far is nanomaterials do bad things, right? So we have to worry about them. The question is, can we make it better? And one way we can make it better is because as chemists, we have the ability to actually make materials with different chemical compositions and sizes and shapes. We can actually then do things that will make them better. And one thing we can do is use advanced computational methods. So everything I've done up to this point is experimental, but computational technology, especially as we go into this AI realm, where now it's possible for computationally to predict what happens with these kinds of chemical reactions. So we're beginning to take steps in that direction of what we call computationally driven research. Okay, so I'm not going to talk about that, but the important point is there is hope that we can actually turn the corner and not just worry about understanding what makes things bad. We can actually focus on how do we make things better. Okay? I did want to talk a little bit specifically about Joel's emphasis as part of this work. So again, there's many different aspects to the work, and Joel was particularly interested in this question of, well, how does the nanoparticle get into the cell in the first place, right? And he was very interested in this idea of the cell membrane. So a cell membrane can be modeled in the laboratory using something called a supported lipid bilayer. So these are two molecules thick. It's basically the barrier at an outer periphery of the cell, 2 molecular layers thick, and it can be sometimes ordered and sometimes it goes into a disorder. There's all kinds of molecules in the cell membrane. There's things like cholesterol, there's other things that change the way that these molecules organize. And Joel was interested in this question of, can we understand like how the nanomaterial actually interacts with the supported lipid bilayer? So these lipid bilayers have different regions, some of which are ordered and some of which are disordered, as over here. And the disordered ones have a little bit different height. And we can probe that using something called an atomic force microscope. So here's an image where you see these little regions here that are higher than the regions surrounding it. These are called lipid rafts, but they're regions where they're ordered rather than being disordered. Okay? So if we take that and expose it to nanomaterials, you can follow in time. You can essentially make little movies that show you how this ordering of the molecules is changing as a function of time, right? So this is work that Joel was really very, very, very heavily involved with, is trying to understand these processes of how does the presence of a nanoparticle influence this organization of molecules within the cell membrane. Now, again, our goal is to connect very fundamental science with what happens in an organism. And I've emphasized that's a really hard chain to draw, right? But Joel was very, very central to our efforts to do this. And this is an example where Joel and a postdoc, Ariel Mensch, did these studies. This is done in a laboratory using simple, a bilayer. This is a model system that's made in the laboratory. It's made on a piece of glass. It's not an actual cell membrane. So we can see these things in the laboratory. The question is, how do we know that these are actually relevant when we actually have some kind of complete organism? Well, Joel really wanted to make that link. And so the way he made that link was to do some studies using some trout gill cells. Why trout gill cells? Well, if you think about an organism like a fish in an aqueous environment, you know, food comes in through the gills or they breathe in water through the gills, that's how they get their oxygen. And the gills can then trap nanoparticles, so they would be potentially like the first line of defense. So here again, you see these images where you can see these ordered regions and disordered regions in actual cells. And then this is kind of a hard image to show, but this is a microscopic image where there are different cells. The cells are delineated by the lines. So the blue things is the nucleus of each cell. They're not all in the image. But then he exposed them to these nanoparticles and was able to look at separately at the regions where the lipids were, where they were ordered, where they were disordered, and where they overlapped. And it's very hard to see here, but the important point is you can see regions where they're green, which is where you have the lipids. You have regions where there's red, where there's nanoparticles alone, but there's regions where there's yellow. Yellow means you have overlapping green and red, and that means he's seeing preferentially the nanoparticles going to these order domains. It's what we saw in the laboratory, but now actually in real cells. Okay, so example of kind of how he did that. I'm going to skip over this in terms of time. So this is a little summary of the first part. I have a shorter second part of the talk that I'll try to speed up. But you know, the important part here, I think, is that we see that these engineered nanomaterials are used, right? The idea that we use materials in, say, lithium-ion batteries for electric vehicles overall enhances the sustainability of our planet. But we do need to be concerned about the downstream effects and what happens at end of life of these materials and recycle. Make sure that we really put in place recycling. Kind of a goal of what we've been focusing on is this idea of what I'll call design rules. That is, because there are so many different nanomaterials and so many different kinds of organisms in the environment, we can't test every combination. We have to pick and choose. But importantly, if we understand at a really fundamental level, what are the kinds of things that happen? What are the kinds of chemical reactions? What are the kinds of ways that nanoparticles can get into cells? Then we have a hope of developing design rules that allow us then to make safer nanomaterials. And the goal in the end is to be able to achieve the function that we want. For example, materials that store energy, materials that emit light, materials that catalyze chemical reactions, but to do so in a way that we do it using materials that have less adverse impact on the environment. Now, up to this point, I've kind of focused on the idea of how do we stop nanomaterials from doing bad things? Yep, okay. And what I want to then say is we can use these same concepts. And in fact, where the center has gone the last couple of years has been in a rather different area, which is global food security. And the idea that global climate change is causing plants in particular to be susceptible to many fungal diseases, okay? It's kind of the same problem. How does a like what we can do is through the center, we made observations that nanoparticles can play a very positive role as a way of delivering micronutrients to plants. So for example, copper, manganese, many of these elements are micronutrients. It is they go into enzymes, metalloenzymes. These metalloenzymes often are antibacterial, so they can improve a plant's ability to ward off disease. It's like a vaccine for plants. Instead of trying to say, if a plant is susceptible to disease, do we attack the disease? Or do we stimulate the plant's ability to ward off that disease by improving the plant's inherent health? Okay, and this is the latter approach. So a lot of the approaches being taken now in the field of agriculture are based on this idea of kill the disease, right? And a lot of the applications are wasteful, but it turns out that things like copper-based nanomaterials, I'll just show you an example here. So here's the experiment. One can take a seedling and immerse it upside down in a suspension of nanoparticles in water, take that nanomaterial, let it dry upside down. Oops. Plant it in a field and then come back weeks later and characterize how has that plant done compared with a plant that didn't have this treatment done. And this is a busy graph of green and gold. I think Green Bay Packers every time I see this. But the important point here is that this use of nanoparticles as a way of achieving improvement in plant health is real, it's dramatic, okay? That we can, for example, using copper-based nanoparticles, improve the ability of a plant to ward off fungal diseases using 100 times less copper than the conventional treatments that are being used today. So this is something that we are now like focusing on, okay, if this is an effect, how does it depend on things like the chemical composition of the nanoparticle? How does it depend on the shape of the nanoparticle, round versus sheath versus other things? Here's some examples of a plant leaf with these nanoparticles on. We can see when we form two-dimensional, like sheet-like nanoparticles that they like to adhere. Whereas if we have round particles, they don't like to stick as much. So this idea of the initial interaction of the nanomaterial with the plant leaf is a critical part of that. And this is something that Joel was focusing on as some of the last experiments that he was involved with before he became ill, was trying to understand if we have a nanomaterial and we want to understand how does it interact with a plant leaf and how does it depend on the properties of that nanoparticle, how does it depend on it? And I won't explain it, but basically that was an important part in kind of Joel's, some of his almost final experiments as part of the center. Joel was remarkable in many ways. In addition to his science, he also was a firm believer in some of the diversity, equity, and inclusion parts that the center has had a large focus on. He was a great communicator. We have a blog for public science communication. He was a firm believer in that. And he just was an absolute full participant, a remarkable person in every way, smartest person I ever met, and a real joy to have him part of the center. Everything I've told you today is part of a large collaborative effort. Joel was a big part of it, but many other people too. So I only list the other faculty members who were involved here. This work is funded by the National Science Foundation. I think all of you know the NSF and science in general is under an incredible assault right now. Not only the DEI efforts, but the entire idea of doing scientific research is very much under attack. And I want to point out that that money that comes through these grants, the vast majority of it goes to training the next generation of scientists. It almost all goes to the support of graduate students and training them how to be effective scientists. So it's not waste, it's not graft, right? Science, it's our future, really. So just, and of course, yeah, Joel was a remarkable person. I loved working with him. And with that, I'll simply thank you for inviting me. Thank you for listening.
Take a question.
Yeah.
Does graphene and/or carbon microtubes have a role to play in this and a role beyond the chemistry, but more in chemical transport?
Could you repeat the question, please?
Yeah.
He asked if graphene, so graphene is a two-dimensional sheet of carbon atoms. And he asked if graphene and carbon nanotubes have a role in mechanical transport.
And a mechanical transport,
Like
or
what-
whatever role it might could be.
Oh, yeah, and graphene in particular, graphene is two-dimensional sheet of carbon. There's a tremendous interest right now in materials that are sheet-like, all right? And graphene is one example. Graphene is electrically conductive, but it can be optically transparent. So it actually is being used on the front of computer laptop screens as one example, where you need a so-called transparent electronic conductor. Okay. Graphene has lots of, actually graphene is also being used in like lithium-ion batteries, where you have the electrodes used in lithium ion batteries have what I'll call the active material, like lithium cobalt oxide. That is put into a more complicated matrix of material that needs to be electrically conductive, and graphene, because of its very high conductivity, is actually incorporated there as well. So many applications of the graphene material.
Thank you.
Yes,
At one point, you mentioned Joel's connection to prions.
yes,
Which we know are related to Creutzfeldt-Jakob disease.
yes.
Can you give us more information about that?
I actually don't know that much about that part of Joel's research. I will say that there's a connection to the center in that part of what Joel was doing also that I didn't talk about was looking at the idea that in the environment, nanomaterials do interact with different proteins. And there was concern about whether that interaction would lead to persistent deformations of proteins, which would be very much like the onset of these neurodegenerative diseases. Again, I'm not a biology guy, but there was a thread there that Joel was working on. In fact, he has some evidence that in fact, there was some evidence for such persistent things, but unfortunately that work never really became complete. So, yes.
You talked about how the nanoparticles can be harmful to us in the environment, but also they can be good in the sense that they ward off fungus.
Yes.
Are there any people in your research enterprise who are looking at the fact that nature may be adapting to the fact that we're producing this stuff by figuring out how to eat it and turn it into something useful?
Actually, there's a really interesting aspect to that, which was my collaborator, Aaron Carlson at Minnesota, has shown that I showed you that these bacteria are actually killed, or at least the mortality among these bacteria that are exposed to these LCO materials, the cobalt. She showed that if you take the ones that survive and put them through several replicates, what you find is there are now a class of bacteria that actually have mutated and are now resistant to exposure to they can tolerate enormously high concentrations of these cobalt materials and survive. So that's really interesting from two aspects. One is there are some connections between that ability to develop resistance to cobalt. And she's looking at the connection between that and the pathways to conventional bacterial resistance and whether there are some commonalities in the mechanism. I've sort of been interested in the idea that we know that one of the challenges in recycling of e-waste is it's really chemically intensive. It's not that we don't want to recycle, but the very process of recycling is itself environmentally damaging, using large amounts of acid and other things. So I've had this idea that maybe you could imagine having some kind of bioreactor, in which case you could let perhaps these highly resistant bacteria do some of the work for you. Now, that would be very cool, right? But yeah, that would be like, you want to start a little company, right?
As long as they don't come after our car batteries.
Yes, yes, well,
Thank you.
sure.
Bob, what do you, oh, go ahead, Steve.
Seems like there's kind of races going on here. I mean, the environment's changing, so the climate's changing so
Yeah,
fast.
yeah.
And you're trying to adapt to that. And first of all, to understand what the heck is going on.
Yep.
And then second of all, to enhance the positive things and eliminate some of the negatives. Who's winning?
Can you repeat the question?
Oh, the question was that, like, the things I showed with the agricultural area, that the environment is changing so quickly,
Yeah.
that are we able to keep up in terms of being able to make these changes quickly? I don't have the answer to that. I will say that my collaborator in the nano agriculture field is a guy named Jason White at the Connecticut Agricultural Experiment Station, and he's like the world's expert in this field. He might even be on the line, I'm not sure. But, I mean, there's just tremendous opportunity there, you know, we'll see, can we keep up? I don't know, it depends on whether we can slow down CO2 emissions, perhaps as much as anything. The way things are going,
Well, and politics plays a role in it
yeah,
as well.
oh, yeah. Yes, unfortunately, the last few weeks, we see too much of that. But yes,
Yeah, I assume from your presentation that there's no regulatory environment in terms of introducing nanoparticles into industry. It's just sort of after the fact when we find out something goes wrong.
yeah,
Am I
like
correct?
TSCA, the Toxic Substance Control Act, actually doesn't make distinction about size. It's more about chemical composition. So I'm not an expert in this area, but my understanding is that there aren't a lot of restrictions on use of nanomaterials in. Now, when you come to food, all right, I think it's different, and that's one of the things that we don't yet know. You know, I showed the examples where exposing these plants to copper improve their ability to resist disease. It also improves the amount of fruit that they make. We don't yet know whether there are residual nanoparticles, for example, in the fruit, and whether that might pose some kind of potential risk. So far, the evidence is no, but, you know, there's certainly, we need to investigate all these kinds of possibilities before going whole hog. And, you know, we're fortunate that the Connecticut Agricultural Experiment Station has greenhouses, but they also have like true farm fields where we can actually like test what happens in an actual natural environment and not just some simulated laboratory. But I found it remarkable that this idea of a seedling immersed in suspension of nanoparticles and then planted in a field 8 weeks later still knows. And it's pretty, I mean, if you think about it, it makes sense that the usual treatment for resisting these fungal diseases is to spray on a solution of water soluble copper, most of which goes down into the soil, right? If the nanoparticle sticks to the plant leaf, it's like a controlled release. It will stick there and it will dissolve slowly. And so instead of getting one giant dollop of copper, you now get it over an extended period of time, right?
Sure.
And it's like an antibiotic that you take over a period of 2 weeks, right? So,
Does GMO come up in that topic? GMO corn.
not a lot here. The European community, I think we see much more about genetically modified foods and what happens there. Yes, it does come up. You know, this area of research in the US, we are like the only center that's focusing on it. Europe has a much more active group focusing on like food safety than the US does in reality.
I have a really simple question, and that's regarding Magic Eraser and that Mr. Clean product.
Yeah,
Is
which is
that
great.
pardon?
Which is great. It works great.
Are those nanomaterial
I
or,
don't know, actually. That's a great question.
I thought I heard they were.
Are you worried about exposure?
No, I'm just thinking that that would be an example of it, you know, that we're using in our homes.
Yeah.
Well, that slide you had before about DDT, ozone hole, and PFAS is a really powerful slide. Each of those DDT and chlorofluorocarbons and PFAS were introduced to sort of solve a problem.
Yeah.
And then it turned out the problem they created was enormous. But now you know things. So what role is your center going to be playing then in impacting the use of nanoparticles worldwide?
Well, first off, my center's ending in August. Because our 13-year of funding is over and there's by law, we can't renew anymore. I will say that most of the people who I'm collaborating with within the center are very enthusiastic about this field of nano-enabled agriculture, seeing it as a very positive one. The thing that scares me is people who talk about solving the global climate change issue by putting nanoparticles into the atmosphere. That seems like a really bad idea. But I think this area of the nano, of the, I mean, one of the challenges we face is, in order to really make progress, so our center was really founded on the idea that we need to understand at a fundamental scientific level what's happening. And maybe pointing the way toward things that could be better. The challenge, though, is the people who actually use the materials, they have to agree. For example, a lithium-ion battery manufacturer might not want to use a safer material if it provides reduced performance. Okay, and we can't control that, so really all we can do is provide some of the scientific insights that might go into helping people understand what the outcomes of those choices might be. And this is where I think actually the design rules are important, because we have no idea what kinds of materials are on the horizon 5 or 10 years from now. But if we understand these basic mechanisms of interaction that might be common to certain classes of nanomaterials, we can at least provide a starting point that people might know in advance. What's the likelihood of some category of nanomaterial having significant impact?
Do you see something like this electronic waste becoming so big it becomes like PFAS or it becomes like chlorofluorocarbons?
Well, I think we're helped in the e-waste. In the e-waste world, I think we're helped by economy. So about 90% of the world's cobalt comes from the Congo in Africa, which is a politically unstable country. And there are finite supplies. There's very few cobalt resources outside of there. There's some in the US, but it would be very environmentally damaging to do it. So consequently, the cost is driving both the transition toward other materials, but I think ultimately the cost will drive the need to recycle. In that particular case, right?
As long as we stay. Do you see a transition away to earth-friendly?
Well, so the latest generation of car batteries are about what's called 822, about 80% nickel, 10% cobalt, and 10% manganese. But nickel is also not a great, let's hear, it's maybe better than cobalt, but I still think there's going to have to be some kind of regulation. But I think the economy will drive. I mean, Elon Musk has a large recycling plant that is going in place next to the giant Tesla factory. So the economy, I think, is beginning to drive that. Not going to say any more about that.
Any more questions for Bob?
Can you tell us about the continuation of Joel's work among his students?
Yeah, well, I would like the closest of Joel's work in terms of I mean, when Joel became ill, many people within the center stepped up. In particular, a collaborator named Juan Pablo Geraldo really stepped up, and Howard Fairbrother and Rigoberto Hernandez and Zev Rosenzweig. The latter three were all in Baltimore, so they actually helped out a lot. In terms of how that specific work is being continued, in particular, our collaborator Juan Pablo Geraldo is very much along the same lines, so that will continue. What is less certain right now is our funding's ending in August. And given what we're seeing on the federal funding horizon, I have no idea what's going to happen to any kind of federal funding, especially anything using the word sustainability. We'll see.
Thank you.
Well, here's to a future that in which sustainability reigns supreme.
Yes, thank you.
No hurry.
Thank you, everyone.
Thank you.
Thanks.
Yeah.